A bit of housekeeping first. While many of you are familiar with our virtual presentations on the Zoom platform, I'm going to go through a few housekeeping items. Please keep your microphones muted and videos off during the webinar. The mute and video controls are located on the bottom left-hand side of your screen. Also, please feel free to type questions for our presenter in the chat at any time. The chat button is located on the bottom center of your screen. We will answer as many questions as we can as time allows. If you have any questions or difficulties with Zoom technology, you can type your question into the chat or alternatively email Scott Royce. Their email will be posted in the chat. If you are having difficulties with your audio, the call-in information will also be provided in the chat. With that, today we have one speaker. We are going to hear from Dr. Damon Smith, our UW-Madison Extension funded faculty, professor of epidemiology and plant pathology and management of field crop diseases. And he will be filling us in on updates and new developments in the world of crop disease in this dry year, but I know that disease is popping up. So thank you, Damon, for joining us.
All right. I'm just trying to get. All right. See the slides okay?
Yep, we're good.
Okay. I'm going to move a couple of things around on my desktop and we'll be off and running. So I do have quite a few slides here today. If we don't get through everything, that's okay. I am going to spend probably about 3/4 of our time on corn and specifically tar spot. And there's a really good reason for that. Over the past week, we have had tar spot pop up in the Midwest despite the really dry conditions. And so I want to dig in a bit because I have been getting quite a few questions on why that is. Of course, with all of these presentations, there's lots of information. Your head will probably be spinning quite considerably once you're done here, and you probably want a couple of places to go. I just want to remind you, two good resources are my website, the badgercropdoc.com site, which is more Wisconsin-centric, where you can find information on diseases of alfalfa, corn, soybeans, and wheat. And then the Crop Protection Network is a group. This is a national group I work with. We have lots of pest management information, bridging the gap from diseases to insects and into weed management as well. So I would also direct you there. And on that site, you also find really pertinent for this presentation, you'll find a web book actually on tar spot, and then also a second web book on white mold, of which if I have time, I'm going to try to address that on soybeans as well. So again, a couple of really good pieces of information. So let's talk a bit about tar spot. This is the map which I pulled 2 nights ago. It did not update yesterday, so I left it the same. I do think it'll probably update by the end of the day today, perhaps with a find in another state. I was exchanging some text messages with a colleague earlier today. You can see from this map, the gray indicates where we have found tar spot in previous years or previous seasons. The blue down in Georgia indicates that areas have been scouted and tar spot has not been found. And then the gold are this year's positives that have occurred over the last couple of days. So there's a hot pocket kind of in the center of Iowa. There's a couple of counties in Missouri and also a county over in Kansas now. With confirmed positives. And so this has brought up a lot of interest. Folks were, of course, looking for tar spot, but very curious why, how is it that this is actually popping up, and it's popping up a lot earlier than folks were expecting because of the really dry conditions that we've seen across much of the Midwest. So I want to talk just a little bit about where we're at with the cycle, our understanding of what it takes to drive tar spot, some helpful hints on perhaps scouting, and then we'll dig in on management here a little bit. So just a reminder, tar spot is a what we call a polycyclic disease. So it has many cycles per season, and this is why it's slow to get started. But then once it ramps up, those secondary cycles really kick in later in the season, and that's why we can see it move. And folks will say it moves very quickly and almost overnight, and that's because we get into this kind of secondary cycle format where there's almost this exponential increase in tar spot development. We do know, and I've talked about this in the past, the tar spot can overwinter here in Wisconsin. So we do have an inoculum that can originate from here in the state. It does not have to blow in, although it can. The spores do seem to move a pretty good distance. But we do have locally originating spore loads here. Those usually start in the lower part of the corn canopy. And then we do need rain to move it up the canopy. But what we're learning is that we actually need wet-dry cycles to start to get the symptoms, infection and symptom development. And I'll talk a little more about what that actually means here a little bit. So moisture is really important, but it's also alternating with these extended dry periods. And that's something that's kind of hard to wrap your head around, and we're still learning a bit about it. This disease operates a bit differently than some of the other diseases you might be familiar with, where you're thinking, well, it needs really, really consistently wet conditions. And, we've actually run trials in the past where we've heavily irrigated and had very low levels of tar spot, ironically. And also we've seen irrigated situations where maybe we're a little more light and frequent, where it really gets bad quickly. So there is something about the frequency and then that intermittent wet dry cycling that is important here. So what was known about tar spot and its development? I'm sure everybody's seen this slide. This has been around the circuit quite a few times. Just a reminder, in the mid-90s, the folks in Mexico who'd been working on this quite some time looked at temperature and then moisture components, which led to higher risk of development. And so keep in mind, this is sort of optimal. These are the optimal conditions to really make it increase. You need temperatures between 63 and 72. So it is kind of a cool to moderately warm type pathogen or loving pathogen, although we know that hot temperatures are not lethal. In 2021, we kind of had a similar situation here, although we weren't as dry, but we were very hot, kind of going through the early July timeframe. And then it got a little bit cooler, and the rain started kind of coming, and that's what really blew up the 2021 epidemic. So we can't get complacent about temperature. And then we know moisture, very important. Relative humidity seems to be a really big driver, especially as nighttime leaf wetness. And that's been coming up. I'll talk a little bit about some of our own work and trying to understand this in the US. But that leaf wetness parameter does seem to be kind of important, especially at the nighttime hours where we have that wetness. And that could be related somewhat to the biology. It's not unheard of for that to be a really important factor. This winter, a group led out of Bayer Crop Sciences published a paper, at least it's open access. It's still going through the review process, where they were actually able to do some controlled inoculation experiments. And this has been hugely challenging since the 2018 epidemic. The pathogen, unfortunately, is what we call an obligate parasite. So it really needs a living host to survive. And nobody's been able to sort of crack the code in terms of how do we actually do laboratory experiments up until this point, where Bayer has this paper that's going through the review process. And essentially what they did here is they took spores that was on plant material, and they were able to successfully inoculate young corn plants in controlled conditions. And I won't take you through all the steps, because they did quite a bit of work to sort of crack the code, if you will, in these controlled experiments. But what's really interesting is that they needed kind of consistent leaf wetness. And again, we're talking about optimal conditions here to maximize the amount of disease. They needed overhead misting 10 seconds for every 20 minutes for 5 days initially. And then they had to withhold all misting and all foliar leaf wetness after 5 days in order to get disease development. And that's really interesting. So we have a wet cycle and then an extended dry cycle here. They did do this with some temperatures there. You can see they fluctuated 21 to 18, and then they actually raised the temperatures a bit, 26 to 18 Cs. So we're talking about kind of warmer. We're getting up, a little above that sort of optimum that we saw in that and that work from Mexico. So this was really insightful because in our own work, we were seeing things like this from our field data, and we were sort of scratching our head trying to understand what that actually meant. And then these controlled experiments sort of shed some light on it. The other piece that came around is that the incubation period, in other words, the time from infection to when we actually see symptom development, again, under optimal conditions, is around 15 days. And then from infection to where we actually see the secondary cycle start is about 20 days. And we've seen this in the field where, somewhere between 21 and 30 days is a pretty good incubation period in the field. Again, we don't always have optimal conditions, so that incubation period does get stretched out a little bit. So it was good to see that our work was corroborated by these controlled research experiments. So what does that mean for you dealing with this? Well, we also have to consider climate and what's going on weather-wise, right? So we can take what we know from sort of small plot field work, we can then do controlled experiments, and then we have to kind of look at what's actually happening in the agri-ecosystem to really understand how do we sort of really anticipate this thing. We've been doing some work with, of course, weather stations, tracking weather. We're also using cloud-based weather information to drive some of our modeling work. We're also doing spore sampling. So in this picture here, you can see two types of spore samplers that we're actually utilizing. So we're trying to understand when the pathogen that's actually flying through our cornfields. And we're using these two pieces of equipment because one is what we call a passive. So it's just sort of picking up what's actually coming through. And then this one in the front here, this fancy one with a solar panel, this is actually a volumetric sampler. So it's actually sucking 16 liters of air per hour, and then impacting those spores on a small tube. And we can actually quantify how much DNA of the fungus is actually on those slides. And so we can better pinpoint when the inoculum is actually present in those fields. We can then look at what's going on historically with weather and start to put all these pieces of the puzzle together. And some of you may have seen this. I've been talking about this past winter. This is data from DNR, just looking at temperature departures and also precipitation amounts compared to, data going back to 1950. And essentially what we're seeing, and this is just documented data that you can find out on the internet, from the DNR, is that our temperatures are rising. So we have warmer temperatures on an annual basis on the order of about 2 to 3 degrees Fahrenheit. And when we have warmer temperatures, we're getting more air or more water aloft. Okay, so warmer air can hold more water. And then, so that raises the humidity. And then we're also seeing heavier precipitation events or more frequent precipitation events. So the upper graph here over in the right-hand side showing, higher rain amounts in recent years. And then more importantly, especially for this particular pathogen, we're seeing heavier rainfalls. And so the days of, having a week where we might get nice, slow quarter-inch rains every other day, those sorts of days are gone. What we're seeing more of now over the last decade or so are these really heavy rain events, 2 inches or more in a 24-hour period, and then it'll be dry for a long period of time, right? And so this is where this wet dry cycling starts to really become important as we're trying to understand how does tar spot get moving. And we can kind of just look at this anecdotally. So these again are just snapshots of maps off the DNR sites, looking at in the top here, we're going from June through September, and we're looking at the monthly average temperature. More red means higher temperatures than average. And then in the lower panel, we have the accumulated precipitation over the month from June all the way to September. And this is 2018. Okay, so that was the first year we had really, heavy tar spot. We had a dry spell in July here. You can see that second map here, dry, maybe a little warmer, and then we got lots of rain in August, right? So we had this extended dry period and then wet. If we look at 2019, wasn't really like that. We were more kind of consistently wet. In August, we got dry. September, it got wet again, and we did see a late onset of the disease in September. But by then, yield was determined at that point. We didn't see much impact, and the levels really were pretty low because the corn crop basically matured and beat the fungus to the finish. Same thing in 2020, kind of more consistent rain events there, a little bit of tar spot around, maybe from this late August dry spell, and then the onset of the rain, but not really the train wreck we saw in 2018. And then 2021 rolls around and we have wet, dry, wet again, and then really extremely dry in September. And we all know what happened in 2021, especially if we look at the central band here. So this sort of dry here, then really wet, and then extremely dry across the center part of the state. That's really where the epicenter was in 2021, where we saw high levels of tar spot. And then, of course, 2022, not quite as bad, but again, some wet, dry cycling. So we've seen these patterns set up now over the last 5 years or so, and our modeling work is really starting to hone in and pick up on this. And then, like I said, it was corroborated by the bear work, where you have to have some consistently wet conditions and then dry conditions really to start to fire things up. So what does our modeling say? We do have quite a bit of work that's being reviewed right now in the journal Nature scientific reports. We made it through the first iteration of reviews there. Reviews were quite positive. So the idea there is the work is very sound. This is publicly available. I have the link and the citation in the bottom part there. What we've been doing is we have had multi-state trials. We actually have 246 site years of data, which is comprised of both small plot and commercial fields. So we actually have 588 individual observations that make up the data set. And you can kind of see where these dots are. The blue are commercial fields, red are small plot trials around the Midwest, especially in the tar spot centric part of the Corn Belt here, where we've looked at the onset and then the increase in tar spot over time using repeated observations of disease development at these particular locations. We can then take our weather data and align that with the disease information and actually start to build out models, which then we can use for disease forecasting systems. And I'm not going to take you through a bunch of math, but I do want you to know that we have several different types of working models. So we have mathematical equations actually explaining this. I know this slide's a little bit confusing here. What I want you to know is that this first column here is accuracy. So we get challenged a lot on our accuracy of our modeling and how we come about these mathematical models. Do we just sort of turn our brain off and let the computer do the work? No, that's not what we do. We take all this information, the bear work, our previous work, what's being published in journals, and we're using that to guide us in terms of model selection. And we're even starting to look at machine learning type of approaches, in addition to our sort of more standard, what we call logistic regression or probabilistic regression type models. And we actually have landed on what we call a multi-model ensemble. So we're actually running two models at the same time and then averaging the outputs together to get our outputs of risk. And then we've compared that against this RF stands for random forest machine learning, which you don't need to be concerned with. And then this AN is another type of machine learning algorithm. And so what I want you to see here is that in our modeling, we're actually accurate about 88% of the time. Yes, the machine learning inches that up to about 90%. If we look at the random forest, but we're right in line there with our approaches. And so we do miss 10 to maybe 15% of the locations, but you can't just sort of give up on these models. I would argue that 88% accuracy is quite good. That's better than a lot of the weather forecasters out there. So keep that in mind. These are reliable models, they're information-based, they're research-backed. We're not just sort of throwing things out there and hoping it sticks against the wall. Just to show you the response surfaces for these two models, remember, we're looking at probability or risk. And so what the models are actually telling you, what the output is telling you in tar spotter, if you're familiar with this tool, is that we've had weather conditions that are conducive for increase in disease. Okay, and we're trying to balance all the information that's out there and what's been published, and we're actually using 30-day averages of relative humidity and then mean daily temperatures in one model, and then we're also using some nighttime leaf wetness parameters, looking at relative humidities above 90%, those hours that were really humid at night, that gives rise to leaf wetness over a 14-day period, and combining that again with mean daily temperatures. And then we're averaging those two outputs together to arrive at our ensembling approach. And this is what's actually loaded now into tar spotter that's currently running now. So we're actually on version 4 of tar spotter. We do spend our winters reiterating and trying to improve those outputs. So like I said, these models are loaded. So in both tar spotter, and then we also run another version. Field Profit is actually a suite of models that includes our white mold models, as well as our corn tar spot models. We even have a turf model in there if you're interested in sort of all things disease. If you don't care about that and you just want to look at corn, tar spotter would be the app there for that. One thing I'll say about tar spotter is that look at the trends over time, the 7-day trend, and you can kind of see these trend lines out of our Field Profit tool here, which is kind of handy. You know, if the trend is sort of declining over time, that probably really tells you got to just be patient. And really, tar spotter, you know, and this corn tool should be used as a scouting tool. We've had high risk. It's probably telling you if you're inside that V10 to kind of R3 growth stage, you should be out scouting, looking for the disease, and then using this tool combined with your own observations to make potentially a spray recommendation. So just use it as a tool. Don't just turn your brain off and start spraying fungicide according to tar spotter. You still have to have some thought process in that use of that tool. Just to kind of put this out there visually on how this sort of works, it really is just putting the disease triangle idea together. So this idea that we have a conducive environment, we have the pathogen present, and then we have, you know, the host out there, and those things are coming together at the same time to get disease development. And so here's just an example of how this sort of gets laid out in the background, and how we are sort of thinking about these tools and all the sampling and things we're doing. This is a 2021 Arlington field. Again, we have 246 of these site years that we can look at. And what you're looking at here are the tar spot risk. So these are the probabilities that we've had high levels of disease. And then we have this low-cost spore sampler. So you can see the discrete windows of time where we actually caught spores. And then we also have our volumetric spore sampler. And what's interesting too about our sampling is that we see different types of data come out of those spore traps. So we kind of have to use them together to really understand when those windows of spore flight, in other words, when is the pathogen actually in contact with the host, right? And so if we take 2021 at this particular location, you can see these two bars down here. This was likely the first time we saw spores move through. You can see we had a nice environment here according to tar spotter, and of course we have corn. And then we can look at when disease actually developed. And remember, the Bayer paper said it took about 15 days for the first symptoms under ideal conditions. Well, in the field, we don't always have ideal conditions, but we can work that out. And so the incubation period, according to what we see in the field, is somewhere in the 21 to 30-day range. Again, if we're a little more conducive, that can get a little shorter. If it's a little less conducive, that can get kind of pushed out a little bit. And then the latent periods we've been observing in the field are somewhere probably in the 40 to 50-day range. So again, when do we get those cycles built up? And my point in this is that we have to use that early scouting and catch this fungus really early, because it shows up so long after the infections actually happen. And if we get behind this, we will get into this exponential increase, and we'll never be able to get ahead of it. And I took a lot of calls in 2021 where folks were seeing tar spot in early to mid-August, and they wanted to spray fungicide, and that was just way too late. you will throw money away in that type of situation. You really need to be proactive, preventative, and catch this thing kind of early on in the process and make those decisions early. So now that we have an understanding of sort of how this thing is moving and how those wet dry cycles are actually pushing things, and we're considering that, you know, this season we could potentially be set up for kind of an interesting year with tar spot, despite the, you know, the perceived dry conditions here. We do need to talk a little about management. I've showed this before, where we have quite a bit of data now showing, you know, the effect of residue versus resistance. Yes, residue management can be somewhat helpful, although the spores can move a long distance. So even though you might take care of residue that is harboring inoculum in a particular field, you can still have spores move in from outside, and you can kind of see high versus low residue situations. Yes, we can drop the severity of tar spot just a bit, but where we get the bigger drop is where we are taking advantage of partially resistant hybrids. And so we don't have immune hybrids, but we do have some partially resistant hybrids. And a lot of the seed companies do know where that resistance is, even though they don't necessarily advertise it. So I would encourage you to push a little bit on your seedsmen to try to figure out what type of hybrid you have. And I would scout those susceptible hybrids first. That's where I'd be putting my scouting efforts in the coming weeks as you start to think about tar spot and tar spot management. We do know that tar spot takes yield. It does it very well. Here's the yield loss data, 92 to 106 day relative maturities, where we're looking at tar spot severity along the lower axis and yield along that vertical axis. And then we, in the right-hand side, we have 107 to 113 day RMs. We can actually solve for the slope of this line and figure out what the yield loss is. And in grain corn, it's about 5 bushels per acre of loss for every 10% increase in the shorter day RMs, and then the longer day relative maturities, it's upwards of almost 8.5 bushels. So this fungus really likes green material. So the longer a hybrid is green, or the longer it's growing, the more opportunity the fungus actually has. And so with longer RMs, we do see a bigger impact on yield because the fungus has more opportunity to sort of hit it hard. We have looked at planting day. You could plant a short relative maturity really early and kind of beat the fungus to the finish, but you do give up a little bit on yield doing that. If you plant a long relative maturity late, you will extend the opportunity of the fungus, and there is potential there for those hybrids to get hit quite hard. So you have to think about this a little bit and kind of balance that relative maturity for your zone, and then think a little bit about planting date. In silage, we've looked at the impacts as well. So I dug in a bit this winter on, I have 10 site years of data on silage hybrids now over a 5-year period at Arlington, looking at dry matter yield. And you can see, yes, there is a significant impact in terms of increase in tar spot severity on the ear leaves and the drop in dry matter yield. And we can actually solve for this line here and kind of work out what that projected yield loss is. And so I did that in this graph here. So if we just look at, say, 10% severity at chopping time on those ear leaves, you know, we're looking at around a 15% reduction in dry matter yield. It's 27% reduction in dry matter yield at 20%, and then as high as almost 40% reduction if we reach 30% on those ear leaves. So, and I've said for years now that the tar spot really, in my opinion, is more impactful on silage corn from year to year in Wisconsin than maybe grain corn. It's a little more inconsistent on grain corn, but we do see some consistent impacts, especially on BMR hybrids here in the state. So you do need to consider that. And I would say if you see tar spot set in early and you have the ability to kind of chop a little bit on the earlier side, I would err that way rather than letting that hybrid get too dry. We've seen issues where if that hybrid really dries out too much, we get, you know, we can't pack the bunker correctly, and we get all these other secondary issues because we can't get all the oxygen out of that bunker as we try to pack it. So adjusting chopping date a bit can be really helpful there. From a fungicide standpoint, we still get quite a few questions there on what are really the ideal products. We continue to run trials here in this state. We continue to run multi-state trials and kind of look at product efficacy. You can find product efficacy ratings on the Crop Protection Network. Those were recently updated a couple of months back, so you can see the really good tar spot products. And if you look at that tool, you'll actually find that you have quite a few options, especially the mixed mode of action products. So anything that has 2 or 3 modes of action in the jug do a really good job. It's really more about timing. And you can see this is my fungicide rodeo from 2022. We're looking at final tar spot severity on ear leaves, greening in the orange bar at around R5, and then yield in the silver bars, and right up there at the top is this Veltima. You can see Delaro Complete in there, used with tar spotters. So these have been kind of more consistent products for us over the years. Mirvis Neo's in the mix in there as well. So you have quite a few choices, really. I don't get too concerned about product choice, again, as long as you got 2 or 3 modes of action there. But I certainly do care about the timing, which I'll address here in a bit. We've also asked the question recently, we had folks interested in potentially looking at 2 spray programs versus the 1 spray program. And the question came up, well, should I use the really new expensive products first and then maybe come back with a cheaper product? Or should I use the cheaper product first and then maybe come back with a more expensive product at the end of the season? Is that crops trying to finish on yield? And we ran a multi-state study for Darcy Talenko and her graduate students. And so there's Wisconsin data as well as other states in this data set. I just borrowed these slides from Darcy. And you can see the treatments we have there: single apps at VTR1, and then dual apps coming in at VTR1, and then following up 3 weeks after that VTR1. So here's the tar spot severity data out of 5 environments that ran this study last year. Here's the non-treated control. You can see the gold bars here. These are the single passes at VTR1. And then we've got the 2 passes with the expensive followed by a cheaper. And our cheaper product we said was Headline Amp, although it's a very good product, despite the fact that it's a little on the cheaper side. And then here in the peach bars, we have the cheaper product Headline Amp first, followed by the more expensive products. And I would argue there's really no difference in which comes first. We saw very similar responses. And yes, maybe a slight drop in terms of severity with some of these relative to the 2 passes, although it's interesting that Headline Amp here, statistically speaking, is not different. Neither is Delaro Complete in those single passes, or Veltima. So, from a statistical standpoint, if we repeated this trial, we would expect Veltima, Delaro Complete, and Headline Amp to still come out similar to the 2 pass programs there. Here's the yield, this is canopy greenness, and you can see, yes, some improved response, but again, those really good mixed mode of action products still right in the mix. Mirvis Neo is now in the mix there. Headline Amp maybe drops out just a little bit on overall greenness. But things that got sprayed doing, reasonably better than the non-treated. And then here's the yield. We weren't able to tease out any really strong differences here, but you can kind of see things that were sprayed at least once in 2022 had better yields than the non-treated control. But it was, maybe about 10 bushels across the 5 environments, and not enough to really tease it out separate from the non-treated control. So, you could do 2 passes, although, I would encourage you to look at the return on investment there. We don't see strong odds of return if we get that single application on in the VT to kind of R3 window. And that's really the sweet spot there. So that kind of bridges this into, this well-timed fungicide apps, and especially well-timed fungicide apps on susceptible hybrids. We've run some trials comparing susceptible versus tolerant or partially resistant hybrids, looking at, in this case, we're going to look at Delaro Complete, but we've done this with other fungicides as well across an array of growth stages. Again, these are just single apps or apps applied using the tar spotter app. And you can see where you utilize a resistant hybrid, you really start quite a bit lower than on a susceptible hybrid. So that's really your first line of defense. And as we think about scouting in 2023, I'd be going towards those known susceptible hybrids first. to scout and look for disease. And you can see the sweet spot really, especially on the resistant hybrid, is in that VT to kind of early R4. Now you're probably looking at R4 going, well, I'd really like to spray there because, I drove it basically to 0. In Wisconsin, be careful with that. That's almost playing with fire because we can't get planes in a timely fashion around here. So what I've been telling folks to do is you really need to make that decision around the tassel time, make that call to the airplane, and then hope the plane shows up by that early R4 time frame. If you have the ability to spray your own corn, that's different. You could really kind of fine tune this, but just be aware, timely application is really important. You need to be on your toes in terms of making that decision early, especially because plane availability can be really, really an issue. Here's the canopy greenness scores. Again, the tolerant hybrids may be edging up just a little bit, and you can kind of see canopy greenness goes up, especially where we're in that VT to kind of early R4 time frame. And then yield, again, last year, not a great year to get a lot of yield response data, but you can see the VT to R4, those definitely edged out the non-treated controls. Yes, the susceptible hybrid, usually did a little better. But again, you have to think about the economics here. Is the trade-off in yield gain that you might get worth maybe potentially having to do 2 passes on a susceptible hybrid in a really bad year? I would advocate no. I would still like to see those resistant hybrids and one really well-timed fungicide put on during that time span. So here's my tar spot take home. You've all probably seen this before. It's you're probably taking screenshots of it. That's great. Keep it on your desk and refer to it. You can, look at fields. I'd be going after fields that are corn on corn no-till, where I know I have a susceptible hybrid. That's where I'd be scouting first. And then looking at my apps and trying to figure out where the environment's been conducive there. Keep that environment in mind, and it's these leaf wetness periods, but they're probably intermixed with a longer dry spell. And so, I'm looking ahead the next week or so, and we're starting to see some pop-up storms come in after this long dry spell here in the southern part of the state. And we could be set up here for a little bit of tar spot. We gotta stay in tune there. In terms of in-season management, those fungicides, yes, are a decent tool. We do tend to get a response. It's usually out of that single application, and your window of opportunity is VT to R3. That's been a really consistent timing for us. And unless it's going to shape up to be a really bad tar spot year, which is too early to tell, I'm not certain we're going to need 2 passes or that late application to help that crop finish out. I think if you can concentrate on getting that one application of a 2 or 3-way mode of action out, you are going to be in good shape and be able to protect that crop. So with that, I'm gonna pause there for a second, see if there's any questions on tar spot that I can answer quickly from the group. Hopefully the moderators can help me out because I can't see my chat bar here, but maybe somebody can unmute and let me know if there's any questions out there. Or there's just silence.
There's no questions yet, Damon.
Okay.
Just somebody said that they haven't sent in samples to a lab, but they believe they saw it since late last week on irrigated fields. Okay.
Yeah, I would encourage, as folks are out there, I'd encourage either sending me a picture or sending that to the clinic. We have our clinic on campus. That would be good to know so that we can start to post that to the national map. We do hide, we don't post GPS coordinates and all that kind of stuff. We do everything at the county level. But it is helpful to start that process. I will say we've seen a lot of bug poop already this year. So you remember in years past, I've talked a bit about frass happens. So frass is bug poop, and you do need to get your bottle of water out of the truck and wash that black spot and rub it real hard with your finger to make sure that that is not bug poop. You won't be able to rub off a tar spot, but you can rub off bug poop, of course. So keep that in mind. We'd love to see pictures or samples. Please please keep us up to speed if you're seeing things out. I did look at some corn today around the Arlington station, and I was unable to find anything there yet, but a lot of our corn is still quite small in the V6 to kind of early V8 range. So we're kind of getting to that growth stage, but not quite yet.
Damon, Kevin Jarek out of Gamey, Winnebago. Sorry, I joined late. Given the variable emergence events we have in these cornfields right now, I can take you to cornfields that are A plus, perfect white picket fence, and then we've got fields where seed sat in the ground for 4 weeks, then emerged. Then we've got seed that's now emerging for a third time here after this most recent rain. When it comes to the timing of BT to R3, if they had to pick the majority of the field and just go on based what the best maturity is, knowing that we'll have variability throughout, what would be your thoughts or recommendations there?
Yeah, well, it'd be a lot easier if you have control of the sprayer. So, which doesn't always happen. If we assume we can dial that spray in and sort of optimize it, and we have some control over that timing, you could potentially push it a little to the later side in that R3. So wait till the majority is kind of getting to the R3, because you'll probably be able to get that younger crop into that kind of early tassel time where you could protect it. So that's sort of the best case scenario in those fields. I think otherwise, I would look at the majority of the plants there. If you don't have control over the sprayer, I would follow what the majority of the plants are doing and target those to try to get the yield the best. Unfortunately, you're not going to, that's the issue with these uneven emergence type situations. But we know, we've done some of that with wheat, where if you err a little on the later side, that's really where we'd like to see it, to let those secondary tillers catch up. And so we can extrapolate that based on work in that crop, and then also in corn here. So again, if I could control the sprayer, I'd err towards that later R3. If I can't, I would just watch the majority of those plants.
Perfect. Thank you.
Thanks, Kevin. Anything else before I transition into soybeans? I think I got a little bit of time here. We'll try to do some soybean work. Anything else?
Nothing in the chat. So yeah, why don't you go ahead?
Okay, I'll proceed into white mold. I'm sure everybody's seen this and probably sick of hearing me talk about this. The cycle for white mold, very different from tar spot. This is a single cycle per season type disease, and the window of opportunity for the fungus is quite a bit shorter here. It's just during the bloom period. So we have to have that canopy closed. The sclerotia, which are sort of like rat turds in the upper 2-inch soil profile, give rise to these little mushroom-like cups, which then eject their spores up into the open flowers. So your window of opportunity for in-season treatment is going to be from the start of bloom to the start of pod. So R1 to R3. If we wait till we actually see disease, in this case, we will fail miserably with our fungicide success. We've run trials, many years now, looking at late applications of fungicide, and we can never. It's almost like we didn't spray at all with those tactics. So with white mold, you literally need a crystal ball. You can't use scouting, unfortunately, for this particular disease. You have to really pay attention to the environment and have a good understanding there. We've developed Sporecaster, another tool that's in the Field Profit suite, or you can find Sporecaster as a standalone tool out there. That is really your crystal ball, and you do need to use it much like a spray tool, unfortunately, instead of a scouting tool. So it's going to be, out there for you to use, again, during that bloom period from R1 to R3 to try to anticipate this environment, again, thinking about the triangle. That tool has been peer-reviewed and validated 3 different times in the scientific journals. I just want folks to know that it is science-backed. We're not just, again, throwing things against the wall and hoping it sticks. And that particular tool is accurate about 80% of the time. So not quite as accurate as tar spotter, but we had quite a bit fewer observations when we built that particular tool, but it's still pretty good. I would argue 80% is still pretty darn good, and we'll probably never see anything 90% or above out of these models, that's really, really hard to achieve. And you could see that even in our tar spot development. Now, in terms of, thinking about optimizing integrated management plans, I get a lot of questions, of course, on fungicides, especially at this time of year. We continue to run multi-state research trials. I'll show you 2021 data, and then I'll show you some 2022 data. We're actually going to look at a 5-site year data set in 2021, but I'm going to hone in on the combination of Wisconsin and Michigan locations, because we had really high levels of white mold in both 2021 and 2022 in those particular locations. You'll see here along the bottom axis are some of the common products we're dealing with here. Endura continues to be sort of the standard. We do include a 2-pass R1 followed by an R3 application of Endura at the 8-ounce rate. And I would not recommend that, but we do it to basically say what would the best case scenario look like, so that we call that our positive control. And then of course we have a negative control with a non-treated down here. So everything inside the green box is like the best case scenario here. So you'll actually see Endura applied with Sporicaster actually doing just a little bit better numerically. And that was a single application at R3. And then you can see, interestingly enough, Omega, which is a contact fungicide, put on using a 360 drop nozzle, an under canopy type application technology, doing quite well. So remember, the spores go up and kind of hit the bottom of the soybean canopy, and then they're kind of redispersed in the center part and the lower part of the stems. And so the idea here is that late R3, if we can actually put the fungicide on in kind of the middle part of the plant, we might boost efficacy. And you can actually see that a little bit there with Omega. If you put Omega on over the top, it tends to drift out down here in the middle of the pack a little bit. Anything in the gold box here, this is not statistically different from the non-treated control. We also need to keep in mind that where we control white mold, we're actually reducing inoculum return. What does that mean? That means the amount of rat turds or sclerotia that we're actually returning back to the soil when we run our combines through is lower where we actually protect the plant. So again, here are our treatments, and we can actually estimate the pounds per acre of sclerotia actually getting returned at harvest time. And so, we're talking about over 1/2 pound here in the non-treated control versus virtually none where we control it inside the green box here. So also, in addition to yield, we get some benefits in terms of future crops by actually reducing the inoculum load over time where we protect the plants from the white mold fungus. And then, of course, here's the yield. So the trade-off is pretty clear, even though maybe the statistics don't always pick it out, but you can see where we use Endura, we get really nice yields. Where we have the non-treated control or some other things, sometimes they struggle a little bit on yield, although in 2021 that wasn't statistically significant. We look at 2022, this was a phenomenal year for us to get data. Again, I'll focus on the Wisconsin and Michigan locations together. Here we had some of the same treatments. We also looked at some other treatments in here, including our Omega applied with the 360 drops, and then looking at our Sporicaster tool. Things inside the green box here were the best. The non-treated controls inside the red box, anything with an A was statistically not different from the non-treated control. And we do have heads up included in here as well, and you can kind of see where that laid out. So we have a pretty sizable middle of the pack here, and then a smaller best case scenario A group, which includes our positive control Endura at 8 ounces, R1, R3, and then Endura applied with Sporicaster again. And then Omega put on with a 360 drop, either just at R3 or using our Sporicaster app, actually did quite well. So again, trying to think about getting it below the canopy and applying it where it needs to protect the plant. And this was a great year to see strong yield responses where we control the disease. Yes, this was a heavy disease environment, so yields did struggle a little bit overall, but you can see some phenomenal returns here. Where we control the disease. We're talking about 15, almost 20 bushel per acre increases with these particular treatments. So quite positive on the return on investment where we get it right. Yes, Endura remains sort of the The standard, especially that 8 ounces, do not go below 8 ounces on the rate. I get that question every year. I did a lot of rate work with BASF in past years, and 6 ounces of Endura just does not work by itself. So you have to do the 8 ounces single application. I do like that R3 timing. If you don't want to use Sporecaster, it has been a really good timing for us over the last 4 or 5 years. Other things to think about, I know that the crop's already planted, but moving forward, you know, as you look at fields this year, thinking about row spacing, 15s and 30s, or more importantly, I like planting populations. You've probably all seen this data before, but I just want to reiterate that, you know, our extensive multi-site year data, and we've published and talked to Sean about this. We really like, you know, the planting population idea and trying to reduce the planting populations. You could stay in the 15-inch row spacing. I think it's just fine. You can see here, yes, we do tend to get a little more white mold in a 15-inch row spacing, but we can offset that higher white mold, kind of maintain our yield if we're using a modern genetic variety with, you know, reducing that planting population a bit. So we want a modern variety because we want it to branch in those lower seeding populations. And I wouldn't be afraid to drop 110 or 120,000 per acre. We've seen good results there and a good return on investment where we kind of get that dialed in. You could even run a seeding rate study in future years on heavy white mold areas and kind of dial that in for you. And you can see the yield data there, just a bit better on the 15-inch row spacing. So we're trying to balance that yield with reducing white mold. If you don't have white mold problems, I wouldn't get too concerned about this. Keep doing what you're doing, 140,000 on 15s, and you know, but be careful on heavy white mold fields. That's where you got to start thinking about other things from a cultural standpoint. And we also looked at planting populations in high fertility situations. So, you know, for us in Wisconsin, those are heavy manured fields, you know, that obviously the response of nitrogen that the plant has really gives us a robust canopy. And then we see the planting population becoming even more important. So we just published on this here in 2023, looking at base soil nitrogen versus high soil nitrogen environments. And we get a nice response at 100,000 seeds per acre. So we can lower that disease index score where we drop 100,000 seeds per acre. And really the pesticide treatments that we put on top of that really don't separate each other. So we do a good job of lowering that base down, and then we can really choose any fungicide that has some efficacy and further reduce that some more. And really those statistically don't shake out. In the heavier seeding rates, yes, so we start to see some separation. You can see quite a bit higher, especially in the high soil nitrogen. The disease index scores are quite a bit higher. So here's my take-home slide. You've all probably seen this before, but it used to be all black letters. This winter, we took a new look at this, and anything in red has actually been adjusted. So these are new recommendations, some things to think about. Again, like tar spot, white mold control is going to be a multi-pronged approach here. You're going to be trying to look at cultural practices, varieties, thinking differently about how you're planting and managing, maybe using drop nozzle technology, maybe adopting some roller crimping rye. I didn't talk about that here, but we've been doing some work with roller crimping rye with good success on reducing white mold. And then of course, using Sporecaster and trying to just anticipate that weather to make some decisions on what has to be done in season. So with that, I'd be happy to take some questions, but first I do want to just thank my my lab. Here we have a couple of different pictures. My lab's been rapidly changing lately. I've had a turnover of students and postdocs recently. Folks are getting jobs. It's been a really good job market for folks, and so you know, lots of new folks in my lab, but they are the ones that do all the great work there, and I need to give them some credit, especially Brian Mueller, who's my field technician, and Carol Groves, who's my laboratory technician. I couldn't do it without him. So with that, if we do have any time, I know I'm probably bopping up against our end of the hour, but I'd be happy to take any questions if there's anything in the chat.
Yeah, great. Thank you, Damon. So before we get to the questions, we're going to do a bit of housekeeping. If you would like to earn CCA credits for today, Jerry will share the screen with the QR codes you can scan with your CCA app, or there will be a link shared in the chat to fill out a form. If neither of those if neither of those work for you, enter your CCA number in the chat box and we will get it worked out for you. So hopefully that will get shared in a moment here. In the meantime, a question for you, Damon, is what would you recommend for white mold control in double crop soybeans?
Well, I don't have a lot of experience. That's been hit and miss here in Wisconsin. Sean's been trying some double crop beans and you know, he's had a little bit of success, but probably more unsuccess, I would say. I think, you know, in that situation, if you're able to double crop, you're already going to be looking at shorter maturity groups anyway, but we do know that a lot of the shorter maturity groups tend to flower more consistently and over a shorter period of time. And so just getting them planted on time, choosing a pretty short variety to hope to get it to bloom. And then you could look at a product like Lactofen. I didn't talk about the Cobra data in there. That would be a cheaper solution, or even Domark. I don't think I'd be getting too fancy on the double cropped beans unless I was in just a really really bad white mold field per se, but those are some options that are on the cheaper side there. The one thing I'll say about Lactofen or Cobra is that we've been, you probably saw it in the data set there. We've been actually looking at the herbicide application rate, so around an 8-ounce rated a V4 timing, and that's been good in balancing the injury relative to the response and disease reduction that we get. So if you look at 6 ounces of Cobra at R1, usually, at least in my data sets in Wisconsin, we're usually lagging by about 5 bushels from the damage that Cobra causes. What we're trying to do is balance that. The Cobra also, not in addition to modifying the canopy, it actually induces a systemic resistance response in the plant, but that is short-lived. And so what you have to do is kind of get it on at the right time to ramp that systemic resistance response up, but it has to be there long enough for it to protect the plant from invasion from the fungus. So that's the game we play in terms of application. So I think I would go that route, but again, I don't have a lot of firsthand experience in the double cropping situation, just given our latitude here.
Great, thank you. Any other questions for Dr. Smith? And you were just on time, perfect timing, Damon. So
Great.
Thank you. All right, so that is it for today, unless we have any other questions. Thank you again for attending today's webinar. Please join us next time on July 12th at 12:30, where we will hear from Dan Smith on combine cleaning and cover crops, and Dr. Joe Lauer on corn development and corn pollination. So that's it for today. Thank you everyone for coming.